Welcome to the Conversational Flow, a KCAS bio podcast.
Hello, and welcome to the 30th episode of the Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian Weil, and I've been in the flow cytometry CRO industry for about 14 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, an additional location in Europe, and partner location in Australia, I'm here in Philadelphia with Brian, and our producer Jeremy is in Missouri. So we're all over the map. We're thrilled to have you listening to our 30th episode, and it will be available virtually everywhere. Wherever you choose to find and play our podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing. Before discussing any feedback we receive from you, we're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're exhilarated to have you here and looking forward to a fun episode today.
Thanks, Adam. For this episode, we've got a comfy agenda, or a neighborly agenda, I guess I should say. So I'm excited to dive into this one. In terms of medical research news, the first article I've got is about giving natural killer cells, NK cells, the upper hand in the battle against cancer.
Yeah, and so here we've got a paper essentially outlining a unique or expanded use for IL-15 therapies. So IL-15, back in 2008, National Cancer Institute workshop actually ranked IL-15 as first among 124 agents, including PD-1, with the greatest potential for cancer therapy. Now, the issue is that just treating somebody with IL-15 comes with its challenges. It can be very harmful for the patient. And so what you need to do is figure out a way where you could dose either very specifically or have your targets uptaken in a way that they're more functional to be able to combat cancer. And so here what this group is looking at had used CRISPR to be able to identify a gene deletion that then makes NK cells more responsive to IL-15. So you don't have to add a whole bunch more IL-15 for the treatment. Less IL-15, normal IL-15 still then supercharges the NK cells.
That's it. And just like with all of the cell therapy indications where cellular toxicity and cytokine storm and things like that were a huge issue, this IL-15 toxicity is really holding back the whole field in terms of something that we know would be quite effective. So this type of operation that would greatly increase sensitivity while not leading to the runaway toxicity would be a huge improvement.
Yeah, it would. And so in this, the regulatory gene that they had mentioned encodes an enzyme, right? And so this essentially makes it treatable by small molecules. And so really curious to see where this ends up going. It's still somewhat in the early stages here, but certainly opening up an area, as to your point, and the National Cancer Institute workshop is to something that really could pay big dividends. I mean, come on, when you're talking about it above PD-1, anti-PD-1 therapy, which we've seen really take over in cancer therapy, really would love to see this come to fruition.
That's it. And, in terms of how long this has been really banging around the drug development space. As you mentioned, it's been in workshops for quite some time. The paper was actually first accepted for publication way back in May, and then like every 6 months, there's some sort of giant announcement that this or that will impact it. Every time there's an announcement, I'm hoping that their company's launched, and then it'll be a 3-year timeline from then until the company makes it all the way into the clinic. So here's to looking at 2030 and a potential additional tool in the battle against cancer here.
Right, so giving us a timeline there, always good with that.
Right. Yeah, and then speaking of timelines and drug development pipelines, our other medical research news story is around a new medication from Roche, Gazyva, that just published phase 3 data in the New England Journal of Medicine.
Yeah, so looking at B cell depletion for lupus, and I was interested. We got phase 3 efficacy allegory trial, and it talked about 76.7% of SLE patients that were treated with this plus standard of care reached a clinically meaningful SRI-4 response at 52 weeks. Now, I was interested as I'm reading through this. They talked about a 53.5% response on placebo. And I'm thinking that seems gigantic for placebo. Now, it is standard, it's placebo plus standard of care. Sure. Right. So, I mean, that bumps it up a little bit, but it obviously got me down the whole rabbit hole of the placebo effect. And so there is a paper that was published this year, essentially, placebo effect perspectives in clinical research, unraveling the mystery of placebo effect in research and practice. And it's an update by Prashant Mishra and Shashishank Bhargava. And essentially talked about the impact of the placebo effect, how people believe that it works, where it is more prevalent and everything. So recommend that as a good read if you're interested. I went through that whole rabbit hole before realizing that it was on placebo plus standard of care, because I was thinking 53.5% just seems absolutely bonkers. I know we've talked about placebo effect being or increasing, over the last few decades. But curious on your take on everything. I mean, you don't have to dive into my rabbit hole with me, but as far as therapy and everything.
Yeah, I definitely think that result stood out, right? That placebo is so high. As you say, the standard of care sounds like then it's decent, and also that maybe not many people are on it. Otherwise, they wouldn't really need to use that as it wouldn't be as significant of a placebo there. So I find that interesting, just that for such a big disease as SLE, that this standard of care isn't talked about as different. Because if you take that out, the way that we would have seen this in an oncology trial would be that they would screen for people that standard of care wasn't working. Mm-hmm. And then it would make it a much easier trial, where you would start out from the 46.5% of people that didn't have success with current standard of care. And then if you would look at that would be a 50% treatment of everybody that's left. Mm-hmm. So that would be maybe easier for them to see. But I understand why it would be very, very difficult for SLE in this case.
Go ahead. Finish your thought.
No, just to say that, this is, autoimmune space is huge in terms of flow cytometry, just understanding the presence of cells and the impact that those cells can have. As you mentioned, targeting B cells. Roche has this that's getting through phase 3 now. J&J has another bispecific that I believe just published phase 2 results in January. And we've talked about all the cell therapies that are currently targeting B cells for lupus and other autoimmune diseases. So very interesting area of medical advancement here.
Yeah, and it's funny. You and I went on totally different directions. So, too, when I saw this was a type 2 anti CD20, I wanted to learn more about it, right? So, rituximab is a type 1 anti CD20 and was curious both about, targeting CD20 versus CD19 and then looking at type 2 versus type 1. And so, the mechanism of action is a bit different here with the type 2, less dependent on complement, right? The complement pathway is CDC, than is rituximab, which, you can have a reduction in efficacy going with that pathway. And so it's one of the choices here. And the reason I like this is just my background in having been at Centocor, J&J, doing a lot of antibody therapy, not only monoclonal, but as you mentioned, bispecific and multi-specifics. And so really was interesting to learn more about that. And, just as a reminder, so from people listening, I'm sure mostly familiar with flow and targets, the difference between CD19 and CD20. Your CD19 is more your pan B cell marker. So essentially hitting almost all the stages of B cell development. Whereas, CD20 is more isolated to mature B cells. And so, this is where you have your biasing there, the population that you're hitting with the 20, rather than hitting all stages of B cells.
That's it. And, when I looked into the current treatment landscape for SLE as well, there are a couple hormones that have sort of broad-based impact that aren't great. There are a couple specific monoclonals that will attack different parts of the pathway. I think there's one targeted more at the interferon gamma specifically. And then people use rituximab, but they're using it off-label actually. This is one of those drug development things where as they validate a specific use, then all of the insurance companies and others switch to, here's this medication that's been validated for this use versus should we use this other thing that may be cheaper but is off-label, so we don't have as much robust data to show will it be effective on this specific case of SLE or not. So this is one of those helpful things for Roche as they're trying to position this as the best option, and any manufacturers for rituximab would need to do additional studies to then show that this is better to get, a new label put onto that drug as well.
Mm-hmm. Yeah.
But exciting things for SLE, and definitely excited about all the different medications going towards the autoimmune space still. There's that push for a lot of investment over the past 2 years. And again, going off that 3-year timer, that means we've got about 1 to 2 years to start seeing the impact of all those investments going on. So looking forward to it.
Mm-hmm. Yeah.
Speaking of investments, maybe we'll switch over that and really get into the neighborly portion. The first neighbor is more of my neighbor. So Altisa Biosciences did close a $75 million funding series for chronic lung disease treatment, and eventually they're going to go after the common cold. And just a hometown call out that Altisa is based in Atlanta, Georgia.
Now, I was curious about this. I did go more into the science aspect of this, but the headline mentions oversubscribed 75 million Series B financing. Curious if you looked that up or what that means. What is oversubscribed?
Yeah, that means that Altisa went out and said, we need, let's say, $60 million. And they need to go find someone to give them that $60 million. So after they presented their business case to all of the different potential investors, instead of giving $60 million, they had enough interest to instead provide $75 million. So that can either mean that you give up more of the company equity than you initially anticipated, or it can mean that you actually increase the price of that equity and increase the valuation of the company. So that's always a good sign for companies that people think that their value is actually higher than their case that they're presenting to investors.
That's interesting. So is there a downside to oversubscribing? Is there like a dilution of the pool of, company stock or options or anything like that?
Sometimes you can give away a little bit more equity than you were initially anticipating. But in general, oversubscription is seen as a really strong positive signal for the company.
Yeah, I can certainly see that. And then, so going along with the science here, so vipendavir, it binds to the viral capsid and it blocks rhinovirus replication. Essentially, the thought is that, rhinovirus is one of those things that can lead to exacerbations for COPD, like you mentioned. And so rather than treating the issue, the COPD, it's more of a prevention there, preventative measure. Would really be interested to see what something like this looks like in the market, cost wise, right? If you're specifically going for blocking cold, the impact isn't like a cancer or something. However, when you are pairing it with the complications, implications of COPD, then it becomes a lot more prevalent of a higher need specifically. And so, what are your thoughts on it?
Yeah, I mean, it's a cool company. Anytime you've got a small molecule, then it definitely decreases the cost pressures overall. And with a target like the common cold, your potential market is huge. Yes, they're focusing on use primarily in really critical applications, but that's just like oncology, where you start out with the sickest patients to show efficacy. Here you show efficacy here and provide a use case, and then eventually you can expand out to healthy patients as well.
Gotcha. And it looks like then they're looking to potentially expand to asthma and other high risk respiratory populations following.
Correct.
So I wonder if, like you said, expanding out, see this becoming something where, you're shortening the duration of a cold. You're talking typically 3 to 5 days, but then you have complications later on. Obviously, you have your cough that continues for weeks after. What would something like this do there? I know I'm going a far field of the COPD and other specific implications here, indications, but I'll be interested in that.
Yeah, anything that can shorten those colds. Cold season frequently overlaps with allergies, but especially after COVID, everybody's always just a little bit nervous. Like every sneeze, you're like, ooh, like is that going to be a bad thing for me? So hopefully that that can be some sort of relief moving forward.
Yeah.
Yeah, and then the next study is around a very local neighbor for us here in Philadelphia, and in fact in Springhouse, our literal next door neighbor. So I was pointing out their building to Adam this morning. Is Third Arc Bio. So you can't see us on the podcast, but we are currently waving to Third Arc Bio across the quad. And they announced a $52 million Series A extension to expand their oncology and inflammation portfolio.
Yes, this is a couple of J&J alums, Peter Leibowitz and Sanjaya Singh. Both I worked with a little bit while I was over there. It's been 9 years since I was there, so a while ago. This was interesting to me in that the mention of a high conviction, exactly what does it mean to be high conviction? And so expected to deliver significant returns, due to either innovation strategy, have strong clinical data, and so here, essentially, signaling strong investor conviction, it's precise immune modulation platforms. And so really bringing the experience that they've had from a J&J and antibodies and multi-specific things of that nature, to be able to leverage them here.
Yeah, I mean, definitely seems like a well-positioned company between having a lot of talent in the local area and now this new funding tranche. I know they've got clinical assets underway that they've announced, so excited to see them expand. And I know we've got new buildings on the campus, so if they need more space, there's places that they can go, which is nice.
And you see too, their lead asset is ARC 101. So it's a T cell engager, right? It's a bispecific, but it is looking at advanced solid tumors. So again, an area where hadn't seen a lot of headway. And you imagine this is where we talk about high conviction. This really would be a fantastic success story if they're able to push in that area.
Yeah, solid tumors continue to be the sort of holy grail of oncology treatments. We've gotten very, very good at treating multiple myelomas and many other types of cancers in the bloodstream and things like that. But being able to address the specific tissue resident cancer is a huge factor and makes it very, very difficult to get drugs to the right spot in the first place, and then immune activity in that spot where it's not quite as exposed as in the blood. So definitely looking forward to them making advances in that type of area there.
Yeah. So what do you got on tap next for us?
In terms of next things, I just wanted to highlight that spring conference season is here officially. So it is March as we record this episode, and we've got all sorts of meetings coming up in April and May where we've announced KCAS attendees everywhere from Boston to Texas to California, some places in Europe. So excited for everybody to go out and meet us at those conferences and hear about all the great things that our clients and everyone is doing.
Yeah, I think we even got Florida. I don't think I heard you say that. So we've got Cyto coming up as well. We're going to have representation there. WRIB.
Yeah.
It's just the team loves heading out to these different conferences. So always scratches that scientific itch.
That's it. Yeah, and that brings us to then sort of the main topic of automation in flow cytometry.
Yeah. So cruise control, flow cytometry and cruise control. Essentially, paper automation in flow cytometry guidelines and review of systems. So the paper goes over 3 different systems. This is published in Cytometry Part B. We've done several papers from there already. Audience is likely very familiar. And so what are we looking at? We've got analysis of the CELMEC SPS sample prep system. This came out in 2021 by Beckman Coulter. We've got the Sysmex PS10 sample prep system, came out in 2019 by Sysmex, and then the BD Fax Duet sample prep system that came out in 2021, again by BD. And so really went through a comparison of each of the platforms and really enjoyed seeing what the strengths were. I mean, we've talked a lot about automation and why would we need automation? Well, there's certain steps that introduce variability. We've got operator, we've got workflow bottlenecks, human error. Still, we've got manual pipetting, staining, washing, lysing, and fixation steps. And so what we're looking to do is try to simplify and standardize those as much as possible. You've got excellent scientists, but when you're pipetting hundreds of steps or more a day, there's still always a chance that something could go awry, right? And so by automating certain parts of that, it simplifies it. It also gives those scientists the ability to think critically about the work that they're doing and even further than they already do. And really enhances process and science, brings it to that next level.
It does. And I appreciated this paper, because it called out all of the automation platforms that I'm more familiar with from my early days in the CRO industry with the BD, LWA, and SPA, so the sample prep assistant and the lice wash assistant, and highlighting that those are really sort of a partial automation tool, whereas really calling out how these could be used as total automation tools. I question, especially with the PS10, if it's really a total automation tool, because it does still require basically a robotic arm linked to something else to really call it that. But appreciate that this really demonstrated the end to end workflow and all the operational considerations for moving through.
So you've used all three of these units then?
I have seen demonstrations of all three: the CellMac, the PS10, and the VaxDuet. I've used the SPA, I've used the LWA, and I've used the TQ Prep that they mention as the old school, the TQ Prep and Prep Plus platforms that they've mentioned as old school platforms.
Mm-hmm.
And it's interesting, like from a flow tech perspective, the like all it takes is one bad experience with any of these things, and they immediately become like suspicious for the rest of their career. So, without naming names, one of these platforms basically failed to pierce tubes correctly after a certain period of time, and so it wasn't drawing out the correct volume for a standard TBNK assay. So then we were concerned about that and switched back to manual reverse pipetting for some period of time until we could like revalidate it. But even after it was revalidated, everybody just sort of cringed at using it because we were a little bit suspicious again because that one time. And then likewise with the Lice Wash Assistant, one time it ate cells, like we put it into the machine, and I don't know if there was a setting incorrect or what. We couldn't find any setting that was different, but for some reason the patient sample just disappeared after we put it into that. So after those types of instances, it just always makes you cringe. So the level of excellence that's needed from these platforms is very high, because all it takes is just that one time, and it basically makes the whole lab remember only that one incident as opposed to anything else.
Well, it's interesting that you brought that up, because even in the paper, they do talk about reliability, and comparing the three units, there was this significant differential. However, if you look at it, the one unit, the BD unit that they had mentioned, had some issues that they encountered, or more issues than the others. They were blowing through samples. I mean, hundreds of samples a day in the BD unit. And it is end to end, start to finish. You basically put the samples in, and then it loads onto the Lyric and, runs the samples. It really, if that is the machine you want to use, you want to do an IVD instrument, it's a great system. It just seemed like when they got to the point where they had been using it for about a year, they were running into more issues. Did, at least from my perspective and reading the paper, seem like they weren't using the other two instruments quite as heavily, and so didn't encounter those problems as much.
I would agree with that, and I would say that that type of full end-to-end automation also changes the paradigm a little bit, right? Like for us, the investment per sample is so high that we take any precautions necessary to make sure that that sample is tested appropriately. And also, we just don't have backup samples in a lot of cases. Mm-hmm. Whereas for the Lyric and the assay specifically that it's set up to do, or the assays, I should say, because I know there are a couple that BD has put on the platform. Retests are usually possible with a reasonable blood volume. So if you get a 5 mil Cytochex tube, you could probably do a retest if you really needed to. And because, as you say, it is so fast, if there was any type of issue with the sample, a rerun becomes trivial in that case.
Yeah, the number of samples that you're able to run at a time between the different units was quite different.
Yes.
The hands-on time, the change there was also something. Each one definitely improved significantly. The CellMac, when you're talking about a lice wash, went from 21 minutes to 2 minutes hands-on time. And then the Sysmex went from 25 minutes to 6. Now, I was curious about why the initial time was different here. You had 21 minutes for the CellMech as a starting, and then 25 minutes for the SysMech. They weren't comparing exactly apples to apples there. At least between the platforms, like within the platforms, they were comparing it, so you can understand the time there. So they weren't also loading on the same machines and all of that. But I think it still bears that there is significant time savings with each one of the platforms.
Sure. And it does really show you the power of controlling the whole end-to-end process, because any type of interruption in the middle of it can significantly increase your total time, right? So even though the hands-on time for the PS10 or the CellMech aren't high, because it requires that manual intervention, there's no way that it can get to the same type of volume as the fax duet that's like attached.
Right. Now, you had actually said that you didn't really buy the Sysmax as kind of end to end. Interestingly, as I was going through, I was trying to think critically to our workflow and which unit we would go with. And at this point in time, not really in the Lyric workspace, right? And so whether or not we need to pair something up with that, really wouldn't be as much of a driving decision here. And so some of the things that were important were the ability to create cocktails, if there are liquid level sensors, audit trails. Now, each one, I will say, had their own sorts of audit trails. But I was actually thinking that for our purposes, that PS10 by Sysmex checks all these boxes, whereas the CellMech doesn't have the capability to do cocktailing, doesn't have the liquid level sensors, and Sysmex actually has a check for expiration dates. Now, you do have to be careful with the platforms and knowing that you're kind of married for much of it into a certain ecosystem of consumables. Some of them allow you to expand out of it. You have to input certain information. It's just not as easy. Like, for example, BD, obviously they're married into the BD ecosystem. And so that stuff is just ready to go. You're using BD antibodies, whatever. It's, lightning quick. But you do have the option of adding in other things. And talk to me about your thoughts on the various platforms and how it would relate to a CRO such as us.
And that's an important note, right? Like, this paper is written primarily from the perspective of a clinical lab, right? So let's say a hospital or even a reference lab where they're running primarily 5 to 10 assays at extremely high volumes. So that's where the automation becomes important. Whereas we are running, let's say 50 to 60 assays at relatively low volume. So if the steps are slightly different and extremely customized, then all of a sudden these washing steps and things like that become much less valuable because they just have to be adjusted to each and every situation that they might encounter. Towards that, from a bioanalytical CRO perspective, as we're looking at really customized solutions, you're right that the PS10 becomes very helpful. Where I know we've been evaluating a couple different solutions for cocktail prep, but if you wanted an instrument just to do the cocktail prep for you, then that's probably your go-to out of these three, because we either couldn't or wouldn't use a lot of the automated features of the other ones.
I think a couple of things too for the CellMech that would probably increase our costs. And again, I want to be clear that we're talking about as applications that we would use. However, and certainly each one would have strengths in other areas. To your point, a clinical for the facts to it. The CellMech has a relatively large residual wash volume, which is something you want to keep in mind there. The other thing is, don't want to use reagents in the system that had already been used to prepare samples manually or had been used in another CellMech system, right? And so essentially you would require 2 sets of antibodies if you're going to do manual prep for that, as well as putting it onto the machine. And so again, some of these things that may be hidden costs that you might not necessarily anticipate up front.
Yes, absolutely. A lot of the automation efficiency gains are predicated on full batches and things like that. So they'll report out the absolute maximum benefits. But if you're running one at a time, then all of a sudden it becomes very, very expensive for some of these runs. But they're absolutely right, though, that the improvement in CVs can be significant. I mean, if you're looking at these CVs of 3 to 7% compared with the potential impact of all the manual pipetting steps, it can really add up over the course of an assay where there might be 5, 10, 15 pipetting steps that could add to variability depending on what type of step is being configured.
Yeah. And you started out where the cost per sample, right? How much is something like this? We go whole hog automation. Where do you see us getting a return on investment? And so, another thing, I'll be honest, I didn't really go into pricing on the evaluation of these units. More was looking for fit. But that's something that also has to be considered there.
Yeah, I mean, for each one of these systems, definitely they should be north of certainly 50K. Most of them are going to be south of 250K. So somewhere in that 50 to 250K ballpark to initially buy. If you're using these all the time, then, for a very busy lab with a small set of high volume assays, the payoff could be as short as 1 to 2 months, and then there would be a no brainer. But if you're using these for very customized situations, the payoff may be, somewhere around 200 to 600 samples, or even higher in some cases. So it needs to be considered as a laboratory is considering automation.
If we were in the Lyric workspace, would you want to have an end-to-end with the machine integrated with it, or would you want to have that separated?
As long as there's an option to run it without that, I think that would be wonderful, because the assays that BD has as IVD approved and linked with the Duet platform are the highest volume ones. So I think if you're going to get a Lyric and you're in the CLIA lab space, it just makes sense to get a Duet if you've got the space to allow it.
Right, exactly. Now, it looks like, too, though, what they were saying is if you've got an assay that's set up, even if the machine is not, the Lyric is not being used, if something is in that workflow, it will prevent you from using the machine. You can't use the machine until everything has gone through the workflow. But I think what you're saying is if you can still use it, say that there's no samples going on that machine that day, you can use it manually rather than having to do it automated.
That's what I would expect. Yes, either manually or with some sort of high throughput sample loader that's not connected to the Duet.
Mm-hmm, mm-hmm.
So that way you could prep all your reagents manually, maybe with the help of the PS10, and then load them on that way.
Yeah. I think it's interesting because right now you see so much advancement with the flow cytometry, the flow cytometers specifically, but then constantly catching up to the process, right? The hands-on blood processing, sample processing. And so bringing those on just continues to bring the whole industry forward.
I think so. And I love this paper again because of the organization of the tables and the clear process diagrams. I thought it was really well done from an operational standpoint to highlight how these platforms could help and how much they can help in some cases as well. So I would love to see this and then see like instrument providers then like use a sort of standardized table to say like, all right, this is why you should go with ours for your specific platform, or this is how we're going to chart an improvement roadmap or something like that to continue going forward. So
now I did have to point out table 5 and the statement, the last paragraph right before table 5. It says none of the current SPSs is a complete walk-away solution, and improvements to the design function and software are certainly still needed. And they have a summary of pros and cons, right? Now the pros, if you look, the first pro for each one of the units was walk-away solution for sample processing.
Yeah.
So not complete, right, to their note, but that's what they're aiming for. They're trying to get it as, and it seemed like the one that was closest to that walkaway solution was really the fax duet, just because of the fact that it is integrated with the lyric.
Correct. Yeah, that's one of those editing things, I think.
I mean, I only picked up on it because it was like you just read that paragraph, and the
Yeah.
next thing you do is go to the table. But I understood what they were coming for, so not criticism there.
Yeah, yeah. And yeah, and then like with any drug label, as you walk through the cons, every once in a while you'll see like a big one. Like sometimes this crashes and eats your whole run. It's like, oh,
That sounds
that and
bad.
headache and nausea, right? Like it's sudden death. But
I will say a total aside there, going back to the placebo discussion, if you get to that paper, they talk about the fact that you get side effects from placebo treatment, right? You see people with headaches and nausea, so on and so forth, which is really interesting. It tells you that not only is the benefit of some of these somewhat driven by the person's state, and they talk about how you present the therapy to somebody, that if you present it really positively, they tend to respond more positively. And whereas you still have negative side effects in placebo. So I know after this, I'm going to be doing even more digging. I've always loved the background on the placebo effect and all the information I could find there.
So you're saying that if these were presented in the same way, we might be able to avoid the placebo effect of software bugs crashing in the middle of a run?
I don't know I don't know if software does that. There was a study about people having heavy metal for plants versus what was it? Classical music, right? And that the plants listening to classical music thrived more. I think you're still talking about a biological system, but maybe the bits and bytes in a computer will listen, and maybe they behave better for heavy metal than they do classical. Who knows? Somebody needs to check this out.
That's it. Yeah. All right. Excellent. Well, yeah, wonderful paper. Congrats to the authors, and looking forward to more and more of these systems getting expanded out to handle both the throughput and the flexibility needed for Flow here.
Absolutely.
Yeah. And of course, all these things greatly improve safety in the lab as well, because the less that you handle, the less that you can accidentally mishandle. So that does lead me to my fan feedback request. What is the worst laboratory safety story that you've ever heard?
Oh, I've got
All
some
names
there.
will be redacted for the protection of the innocent here, but
Yeah, I mean, it sounds like you have stuff in mind there.
of course. You always hear about the people that were like mouth pipetting benzene while they were smoking in the lab back in the 70s or something like that. I've spoken to one person who mouth pipetted. I've never spoken to anybody that was smoking as they did it. But a colleague told me about a pathology lab, again, that will remain secret at this point, but where people were like actively eating like breath mints and would throw like breath mints across the lab, and there were mints like landing in paraffin that you used to like embed tissue samples for sectioning and pathology review. So heard all sorts of things about that lab and the lax security and safety standards that were used to protect the staff.
Yeah, it was amazing. I remember first getting into the lab. I mean, I started my career in 2000, and at that time, people had their benches in their lab, their desk benches in the lab, right next to where you would do your experiments. And so I did have one time where I was doing rat liver homogenization. And what that entails is you take these livers and you blend them up and whatnot. I, that day, unfortunately, had made the horrible decision to have liverwurst for lunch. and liverwurst smelled exactly like the homogenized rat livers. And I
I mean, it
still,
is essentially the same, is it not? Like
it
it's
is, it's, I mean, liverwurst is a liver pate, right?
yeah.
I still do enjoy liverwurst, but I mean, that was a bad choice that day. I had some other stories from early on at the time, but that one just always sticks out. Nothing bad came of, I didn't accidentally mix the sampler or anything like that. But yeah, the days of eating my sandwiches in the lab really are long gone, thankfully.
Goodness. Um, yeah. So looking forward to our fans giving us some good stories of their early lab days, and hearing about how safety has come a long way since back in the day.
Mhm.
Um, and then in terms of wrap up, I think this episode on automation can get applied to like our daily life too, in terms of how we automate our cars.
Yeah, so I mean that's where the title is going on flow cytometry on cruise control, and had a little bit of fun with how did we pair that with this. And so, first off, I got to ask you: do you use cruise control in your car?
I do.
Do okay? All is like an all the time thing.
Anytime I'm driving and I'm going to be going in a straight line at the same speed for more than about 3 miles, I'll probably turn on cruise control.
I will tell you, I've never used cruise control in my entire life. And so here, I'm going to take this a little bit further. So for my birthday, which is coming up, a friend and I are going to do this exotic car ride. And so I'll get to drive a McLaren 720S. And this is a car over 700 horsepower. It's over $300,000. So I'll never own one of these things, right? But it's just really cool to be able to get to drive. And we were talking about whether or not one of these cars would have cruise control. Like, do you really want to have cruise control in a car like this? They do. They have, like, with all the bells and whistles. I mean, my thought was, of course, they would add it. But would you ever use cruise control in a McLaren 720S?
No, I mean, those are different cars, right? Like, you're not taking that on a road trip. Like, I'm not going to see that on the truck stop highway outside of Dalton, Georgia, going in between like Atlanta and Nashville or anything like that. Like, nobody's taking that on a 4 hour drive.
What do you think the gas mileage is for something like this? I do know it, so I'm curious.
I'm going to guess 8 miles a gallon.
It's 15 to 22,
Okay.
which is the same as my car.
Yeah.
But anyways, I digress. So I'm looking forward to being able to go with my friend drive. I think we're gonna have a blast. But you had also mentioned you added a couple of things to
I did. So to go farther than cruise control, when you add in lane keeping assist or full self driving, does that change your response?
I I think those are helpful, right? I think certainly for people who don't have the confidence in driving, having those sorts of things are beneficial. And I don't say that I wouldn't use it. So you said lane assist, and what was the other one?
Full self driving. So that some cars can go from like 60 miles an hour all the way down to zero, so they can come to a full stop on their cruise control.
Mhm.
So for highway, if you're in like bumper to bumper traffic, you could theoretically turn that on and it could drive for you. It could change lanes. In some cases, some cars take exits.
hmm Now, this we had talked about a little bit, the self-driving stuff. And it's interesting because you could provide data to somebody that self-driving, there are fewer fatalities than if you were manually, you're doing it yourself, right? People are like, I want to be able to control how I go out. And so the car is the one that did it to me. I didn't have any control over it. I don't
Well,
know.
and it's the same exact thing. I'm glad we did this automation episode at the same time because, I mentioned people being forever suspicious of the LWA and SPA as they were doing things. But if you look at the manual error rate, it's much higher than the one sample that everybody remembers. Because again, like you don't have that personal control. So it's the same exact thing.
Yeah, it is certainly. So, do you do self-driving? I mean, do any of your cars have that?
No, I do not have a car that's capable of that amount of stuff. I think it's pretty fancy when some of the cars I get in will have cruise control that can slow you down a little bit on the highway. I'm like, okay, this is pretty cool. But I don't have any experience with a car that can do all of it.
You say a car that's so advanced that it has power windows.
No, all power windows. We're set there. I am fully in the 2010s, but have not moved up to the 2020s with the advanced cruise control functions here.
Very nice, very nice.
So,
all right, any other thoughts there?
No. I think great talking with you as always, Adam. Excited to hear more about this automation. And thanks to all of our guests for listening. Looking forward to everybody tuning in on our next episode.
Yeah, thank you very much, everyone.